Okay, so we are going to continue talking about T cells, and importantly, how T cells use their T cell receptor to detect pathogen signals, and how that mode of detection of the T cells, how they work, is quite a bit different than B cells. And yet, at the same time, B cells and T cells have to work together in order for you to get the strong antibody response out of the B cells that you need. So let's kind of think about the evolution of our innate and adaptive system and these two big parts of our immune system that we said, on the one hand, our innate side evolved over eons of being exposed to the same types of pathogenic signals so much that eventually, as part of natural selection, our genes became programmed to be able to code for certain molecules like complement proteins, or the creation of certain cells like macrophages or neutrophils that are very good at detecting common everyday pathogen signals, like the rainbow hair, the white face paint, the colorful outfit of our typical pathogen clown, right? And so for most of our daily exposures, that's enough. And it's good because it takes care of 99+% of the different infections that we encounter on a daily basis. And it does so in a way that's very modest to maintain, not too expensive, right? And it always follows the same sort of game plan. And so that involves cells like macrophages, dendritic cells, other granulocytes like basophils, eosinophils, neutrophils, of course, that come and provide backup, and also natural killer cells, which we will get to in more detail later. So the last few lectures, we've been looking more at our adaptive side. We have an adaptive immune system because since we live for many years, it's inevitable that you're going to encounter pathogens like bad clown here, exotic pathogen, who has, due to maybe mutations or something like that, different types of signals on its surface that are different enough that the innate effector side of our immune system can't do a good job of really detecting him fast enough or thorough enough to be able to eliminate him. And so as a result, he has the opportunity to colonize, spread in our bodies and make us sick, right? So our adaptive system has to have some way of identifying this novel pathogen, spreading the word, and then mobilizing a very strong and powerful response to deal with him, because by then you're probably really sick, so you can't waste any more time. So we said that B cells produce immunoglobulins on their surface with many random shapes, and the hope is that if you have a few 100 million different B cells circulating around in you, then if bad clown comes along, some of those immunoglobulins will detect features on different types of antigens presented by bad clown, and those immunoglobulins then serve as the basis of becoming antibodies that can be released and provide the much needed targeting. Last time we learned that T cells are an important part of this equation as well. So T cells communicate with B cells and tell B cells what to do once B cells have been chosen because their immunoglobulins match up with something. Right? And we said that there are two main groups of T cells that are part of our adaptive immune system: our CD4 positive T cells that are the helper ones that actually help not just B cells, but other cells like macrophages do a better job when the alert or the alarm is raised that there is a novel pathogen, and cytotoxic T cells that will actually go and attack and destroy the pathogen directly. So there is some overlap between the two, right? So there is some functions of T cells that we will learn about. There's a group of T cells called gamma delta T cells that actually function more sort of like in between adaptive and innate effectors. And the natural killer cells sort of occupy this halfway between adaptive and innate. So we'll get to those. So keep in mind, a good way of thinking about it is innate effector cells act on the pathogen directly. Like a macrophage attacks the pathogen directly, like this bacteria, and eats it and destroys it. Whereas adaptive effector cells, right, they don't go after the pathogen directly. This is key. They go after other cells in your body that show evidence that they are in trouble. Maybe because those cells are harboring a pathogen, like a virus or a bacteria, right? So those cells need to be told to eliminate themselves, take themselves out of the picture to stop the spread of the pathogen. So that's a good way of thinking about it. Adaptive effector cells, like T cells, right, act on target cells, not on pathogens directly. All right, so let's compare and contrast, because both B cells and T cells provide adaptive specificity. Both of them carry unique antigen binding sites on their surface, right, in the form of either immunoglobulins on B cells or T cell receptor that give them the ability to recognize a specific target. If they happen to encounter a matching antigen, then they activate. But their effector roles of B cells and T cells are different. Let's contrast here. In B cells, we said chosen IgM on the surface of a brand new naive B cell, right? That IgM is the basis for making antibodies. The very first wave of antibodies that will be made are IgM antibodies, right? Remember, there's 5 subtypes or classes or isotypes of antibodies, M, D, G, A, and E. Since brand new B cells only produce IgM and a little bit of IgD on their surface, then if they are chosen and told to make an antibody version of that, your first antibodies are IgM antibodies, right? So when B cells are chosen, they are rapidly proliferated. So you have this one rare B cell that has a unique Ig that turns out to be valuable. If it's chosen because its Ig works, your body doesn't want to lose that B cell. So it makes many, many copies of itself identically, and all of those identical B cells make that same chosen immunoglobulin on their surface, right? Some of them will then be told to become plasma cells and start making the free-floating antibody version of that IgM, just to get some IgM out there into your system, right? Where the other clones might be told to undergo hypermutation to try to improve the fit of the immunoglobulin to make better and better antibodies as your battle against the pathogen progresses. So here's a plasma cell on the bottom. Here, right, it expresses antibodies. You might be tempted to think that all these little bumps on the surface, oh, those are the antibodies. Well, no, these are just sort of different bumps and things in the cell membrane, because the antibodies are pretty small proteins. You would not be able to see an individual antibody if you could see a plasma cell at this size in the image, right? But if you look on the right, these are individual antibodies, right? So you can see some of them, you can make out the sort of Y shape of them, right? So these are immunoglobulins that are now released by the plasma cell as free-floating specific targeting molecules. T cells, right, they're also chosen on the basis of whether their T cell receptor has the right fit to recognize certain antigenic signals. But remember, T cell receptors, they stay on the surface. They're never released, and T cell receptors are limited to only being able to recognize small fragments of proteins, small peptides that are presented to the T cell by other cells that the T cell bumps into. And those other cells present evidence of what's happening inside of them on MHC platforms, protein platforms on their surface, and those MHC platforms present small little peptides to whatever T cell might bind to it. And the T cell has a T cell receptor that has maybe the opportunity to see if it recognizes peptides. If it does recognize a signal, that T cell, depending on whether it's a CD4 helper or a CD8 cytotoxic T cell, will either help the target cell. Maybe that target cell is a B cell, right? That's getting chosen, and that helper T cell might help that B cell by telling it, hey, you need to divide, make many copies of yourself, and start making antibodies or start hypermutating, or if the presenting cell is an infected cell in your body that maybe has a virus in it, then a cytotoxic T cell might tell that cell, no, right, you need to undergo apoptosis, which is programmed cell death, to take yourself out of the picture to spare the rest of the body the spread of disease. Okay, so this is what we discussed last time, but a quick review. CD8 T cells, CD4 T cells, they both have T cell receptors all over their surface. It could turn out by chance that this alpha beta T cell receptor that you see on the left could be identical to the T cell receptor you see on the right, just because these two T cells, right, they might have made T cell receptors that, by chance, the variable domain of the T cell receptors can bind the same peptide. In this case, the peptide antigen is the red dot that's being presented by whatever target cells the T cells are encountering. So on the left, remember, you have a target cell that's maybe any old normal cell in your body that got infected with a virus, for example. So the virus is being broken down inside the target cell. Small peptide pieces of viral proteins are then presented on MHC1, right, the yellow platform over here. So this CD8 cytotoxic T cell, its T cell receptor recognizes the shape of that peptide in the context of also recognizing the shape of the MHC platform that's presenting it. When that binds, the CD8 co-receptor that's on the surface of this T cell swings over, and that CD8 confirms that this is MHC type 1 platform that's showing off this peptide. And those two signals then, the T cell receptor seeing the red dot and CD8 confirming that it is MHC1, will activate the cytotoxic T cell. It will release certain cytokines that are actually cytotoxic messages, right, that tell this target cell to undergo apoptosis. On the right, we have a professional antigen presenting cell, maybe like a macrophage or a dendritic cell or a B cell, that got peptides or proteins from, let's say, the virus inside of it, not because they got infected, but because they're doing their job, right? They're detecting different pathogenic signals, right? So they're going to process the proteins differently and present maybe that same exact red dot, but on MHC2, right? So remember, these professional antigen presenting cells, they have MHC1 just like every other cell in the body, so they can show off peptides if they are infected and need to be eliminated. But as part of their job, they also have MHC2, so they can show a message off as part of just doing like this job as a messenger, and they don't want to be killed or destroyed by just doing their job, right? So CD4 T cells, their T cell receptors recognize the peptide on MHC2, and the CD4 co-receptor comes over and confirms this is MHC2, right? And so once those two signals activate, the helper T cell will also release cytokines, but these cytokines are not cytotoxic. These are cytokines that will help the professional antigen presenting cell sort of up its game, right? Accelerate its activity and contribute more to the immune response. So the combined signal of the TCRs recognizing the specific antigen and the co-receptor, either CD8 recognizing MHC1 or CD4 recognizing MHC2, is what will activate the T cells by turning on the expression of certain genes in the T cells nuclei, including things like certain cytokine genes. Right. So this figure we looked at, I'm not going to spend a lot of time with this today since we went over it in lecture pretty extensively last time, but it's a nice reminder. There's different scenarios, right? If you have an infected cell that needs to be eliminated, it's presenting evidence of the infection on MHC1, so that this dark purple CD8 cytotoxic T cell, if its T cell receptor happens to be the right one or the right shape to recognize that peptide that's been presented, and the CD8 co-receptor confirms that this is MHC1 that's doing the displaying on this target cell, then the CD8 cytotoxic cell tells that target cell to die. In the other two scenarios, rows 2 and 3 here, right? We have professional antigen presenting cells, a macrophage and a B cell, just doing their job. Macrophage eating things, right? Bringing in evidence as part of its job, cleaning up and detecting pathogens. And we have the B cell binding things on its immunoglobulins and constantly recycling those immunoglobulins, just like any other protein on the surface. They're pulling the immunoglobulins in to replace them with fresh Ig molecules. So anything attached to those immunoglobulins also gets brought in, including, in this case, bits and pieces of the pathogen that then get processed, right? And the B cell will present that on MHC2, just like the macrophage will present evidence of this bacteria on MHC2, so that a helper CD4 positive T cell in light blue here, if it comes along and recognizes that, will send helpful cytokines over to the macrophage or to the B cell, telling it what to do next. Notice the cytokines that are sent over to the macrophage in the cartoon are light blue colored. The cytokines that are sent over to the B cell are these green triangles, right? So hopefully this gets you thinking, yeah, because the messages that need to be sent from the helper T cell over to the target cell need to be specific messages that the target cell understands. In other words, cytokines in the language that macrophages understand, or cytokines in the language that B cells understand. So that means the helper T cell that's doing the, cytokine signaling, right, has to be sort of different. The CD4 T cells that are interacting with macrophages have to be slightly different than the ones that interact with B cells, because they speak a different language, right? So that they can talk to appropriate target cells. We will get into that a lot more in subsequent lectures. All right. So the question was raised, do professional antigen presenting cells also have MHC1 on their surface? I mean, they have MHC2, right? Because that's how they pass the message on to T cells, helper T cells, that they have discovered or encountered some sort of pathogen. But yes, all professional antigen presenting cells, just like every other cell in our body that has a nucleus, also has MHC1 on its surface, because a macrophage or a dendritic cell or a B cell can also get infected, right? Okay. So here are two scenarios. So here we see that a healthy macrophage is phagocytosing or eating bacteria, right? And so that bacteria that gets into the macrophage is always contained in special environments like a vesicle, right? So as the evidence of the bacteria is broken down, right, all that evidence is still contained in this vesicle, and then those parts can be recycled and eliminated. All right? So in this case, the healthy macrophage, the MHC on its surface should only be presenting normal proteins, the normal healthy cellular proteins that are floating around in the cytoplasm and that are also being broken down into bits and pieces. And all those normal self-peptides are what this healthy macrophage are showing on its surface on MHC1. But MHC2, right, is what's going to load up evidence of this bacteria that's being taken in, and it'll be MHC2 that's displaying the bacterial peptides for a helper T cell to come along and see. However, if this macrophage itself got infected, for example, by a virus, right, then you would imagine that the MHC1 on its surface would also be displaying viral proteins, right? So shown here, that's what the glowing red is supposed to show, right? Because this macrophage, if it itself is infected, it needs to be eliminated, right? But at the same time, yeah, its MHC2 is probably still displaying evidence of this virus as well. So in this case, an infected macrophage can communicate both with helper T cells to show the helper T cell what's inside of it or what it's taking in, right, in terms of viral proteins. But more importantly, it can interact with cytotoxic T cells that would then tell this macrophage, sorry, you need to eliminate yourself through apoptosis. All right. So how does this selective loading of peptides onto MHC1 or MHC2 happen? Right. It has to do with where the pathogen peptides are located inside of the cell that's doing the presenting. So consider if a macrophage or a B cell, right, is taking in different peptides or different antigens from a pathogen as part of that macrophage or B cell just doing its job, so they are not getting infected, right? Then those evidence, those protein bits, will always be contained in special vesicles. So it makes sense then that if those protein bits are broken down in special vesicles, right, and that's where the evidence is within the cell, right, not just floating around randomly in the cytoplasm, then MHC2 platforms should go to those vesicles to get loaded up with those peptides to then go to the surface and display. So that's what we see here. So here's a macrophage. Let's say here is a endosome, right? So let's pretend it takes in stuff from the outside, and it forms a vesicle that contains that stuff. Then you might have lysosomes that contain digestive proteolytic enzymes that might fuse together with that endosome to allow for the digestion of different proteins and things like that. And here then is where the class 2 MHC2 proteins, the platforms, should get loaded up with those specific evidence. And then those will be coming from the Golgi, right? Because remember, MHC2 is made out of proteins. So if you backtrack, that means they are expressed by genes that are expressed in the nucleus as mRNA. That mRNA is then probably translated into proteins by the endoplasmic reticulum and then moved over to the Golgi to be packaged up and shipped around as vesicles. So maybe this vesicle over here contains MHC2, for example. So that can join up with the vesicles that contain the evidence of all the little peptides from the pathogen that this macrophage just ate, right? And in those vesicles, the MHC2 will be loaded up and then brought back to the surface so that the MHC2 platforms can now present peptide on the surface. Right? If, on the other hand, this was a virus that infected this cell, then all throughout the light blue cytoplasm, right, you'd imagine all kinds of viral proteins floating around in here, right? So that's very different than having them contained in special vesicles, right? So proteins that are floating around in the cytoplasm, whether they are pathogenic in origin, or most of the time, there's just going to be an abundance of all kinds of normal cellular proteins, your self proteins that are in this light blue cytoplasm, right? Those get shipped into the endoplasmic reticulum, right? To be recycled, maybe folded up again, right? If they are normal proteins that can just be kind of like finely tuned again, put back into their right shape to be shipped out. And all of the pathogenic peptides also get shipped in there. So that's where class 1 MHC platforms or MHC1 platforms get loaded up, right, in the ER. And then they get shipped already loaded with peptide out to the membrane to display. Okay. So I said that cytoplasmic proteins are constantly being recycled by our ER, but also by things that are already found in the cytoplasm. So our proteosomes are these big molecular protein complexes that are essentially recycling bins, right? We have many proteasome complexes in our cells, and these really are like recycle bins. Or another way to think of them is like a shredder, right? So just like an office shredder, where if you need to recycle old papers, you feed them in, and the papers are shredded up into small little pieces, right, that facilitate or make it easier, for example, to then bag them up and recycle them, right? So here you see a proteasome represented as a cartoon, right? So just like a recycle bin or a shredder, it has a cap on either end, the 19S cap, made of proteins, and then these different beta units in the middle that are proteolytic enzyme subunits. So anything that gets fed into the cap, when the cap opens up, right, these proteolytic enzymes can then chop up long proteins into small little peptide fragments. Right? And then those come out the other end, and those are what get then moved into the ER to be further recycled or maybe refolded, etc. Right? During an infection, infected cells can put together a slightly modified form of the proteasome that we call the immunoproteasome. This one has different types of subunits in it that tends to chop up the proteins into smaller pieces than the typical normal constitutive proteasome does, right? And these smaller pieces, the smaller they are, the better they potentially will fit on the top of an MHC platform. Because remember, MHC platforms, as we discussed last time, can only hold small peptides. MHC1 platforms typically only hold peptides that are like 10 amino acids long, right? So if you have a big protein that was potentially several hundred, maybe 1,000 amino acids long, right? Chopping it up into small, teeny tiny pieces of 10 amino acids, that's kind of a lot of work, right? So if you're curious what a proteasome really looks like, because maybe this cartoon isn't very satisfying, here it is. Right? So what we see in the middle, the orange and the yellow is the core part. That's where the core of the barrel is with the proteolytic enzyme subunits. And then the purple and the blue on either end are these caps that can allow for a misfolded long protein to be fed through here to be chopped up. All right. So I said little bits and pieces of chopped up proteins by the proteasome go into the ER, right? And that's where MHC1 has a chance to load them up before MHC1 is moved to the surface of the cell to display, right? So here you see in this figure, let's start from over here. Here's a proteasome, right? The recycling barrel. Here you see a long protein that's sort of unwound being fed into the proteasome. And in the proteasome, it gets chopped up into little bits and pieces, right? These bits and pieces are going to be all kinds of random lengths because the general constitutive proteasome is just kind of chopping things up. Typically, they're chopping proteins up at serines and threonines, right? So you might have fragments that are tiny, you might have fragments that are bigger. These peptide fragments then go to the ER, and they are fed into the ER through these special, in this cartoon, these green proteins that are like a gate that allow these small peptides to go into the endoplasmic reticulum. So let's back up to the first panel here. Right? So in the ER, you have all kinds of proteins that are being made, right? Because remember, ribosomes, as they're translating mRNA into chains of amino acids that ultimately need to be folded up as finished proteins, many ribosomes go to the ER to actually translate the mRNA so that the resulting polypeptide chain ends up in the ER so that you could have other proteins in the ER help with the folding of the protein before the ER then ships these out in vesicles, maybe to the Golgi, for example. So the MHC proteins that make our platforms on the surface of the cell, they're no exception. They are proteins also. So here in the first panel, you can see MHC1 platforms are being put together. So you have two parts to the MHC1. You have this longer yellow protein that is being assembled, right? And then you have this smaller beta globulin part of the platform, right? That also comes and is assembled. This protein, calnexin in the ER, helps with the assembly. So once it's put together in the middle panel here, right, you have other proteins that are helping to really form this into the right shape. Okay, so you don't need to know the names of these for this immunology class. I just want you to realize that this is what's happening in the ER. The MHC1 is being put together just like any other protein, right? Gets help from different types of chaperone proteins and things like that. And now we have these small peptide fragments that are coming in through this TAP, right? Or transporter associated with antigen processing proteins, right? These are the gates that allow these peptides from the cytoplasm to now come in. You have another protein, this sort of violet or purple colored sort of Pac-Man looking thing called ER aminopeptidase, or ERAP. And that edits or trims the peptides down, right, to be more optimal in size, like 10 amino acids or 15 amino acids, so that then they become the red dots that the cartoons like to show, right? The red dots are like 10 peptide long or 10 amino acid long amino acids. Now they can be loaded on the properly formed MHC1. So the MHC1 that's already loaded with peptide in the ER now is shipped out, right, through the Golgi and eventually to the surface of the cell, where now this cell is doing its responsibility of displaying evidence of what's happening inside of it. Most of the time, right, this protein, right, that's being processed is a normal self protein that's just being recycled. So normally the MHC1 on the surface of a healthy cell is just tons and tons and tons of different examples of normal self peptides, nothing that a CD8 cytotoxic T cell would care about. So here's another illustration. I like to show you different illustrations of the same thing because I think oftentimes different pictures help you realize what's going on, right? Proteasome in the cytoplasm here, recycling this protein into randomly sized different fragments, short ones, big ones. They are fed through the TAP sort of protein gate into the ER. In the ER, you have MHC1 being formed, right? So here is a ribosome out in the cytoplasm that was reading this black line here, which is the mRNA that codes for the MHC1 proteins, right? So the resulting orange MHC1 polypeptide chain, the protein is being translated into the ER, where helper proteins begin to shape it, just like any other protein undergoes the same sort of folding process with different partners, right? And then once that MHC1 is ready, then these different sized fragments of peptide get trimmed down by ERAP to be just the right size, maybe 10 amino acid long fragments that can then be loaded on the MHC1. The loaded MHC1 is then shipped out from the ER to the Golgi. The Golgi then puts certain marks on it that sort of label this as being destined to head out to the cell membrane. And then the MHC1 ends up on the cell membrane, where there's an opportunity for a cytotoxic T cell to perhaps encounter it. And if this protein that was being processed through this whole thing is not a self protein, but is in fact something from a virus or from a bacteria that somehow is infecting the cell, then hopefully you have a cytotoxic T cell that has the right shaped T cell receptor to now recognize it, right? And then that's how the cytotoxic T cell now has a chance to see that something is going wrong inside of this target cell. All right. On the other hand, how do we load up MHC2, right? Professional antigen presenting cells that take in pathogen, not because they're infected, but because they're doing their job, taking in pathogen, processing it, and displaying. How do they get those peptides on MHC2? So in this cartoon here, the gray cell represents a professional antigen presenting cell that has maybe eaten, evidence of a pathogen. So this protein now is maybe from a bacteria, it's a fragment of a bacteria or whatever, or a virus, right? So when it gets into the cytoplasm, it's not just randomly floating around, right? It's not going to float around and be recycled by the proteasome and sent to the ER. It's contained and remains contained in special vesicles. In these vesicles, you have proteolytic enzymes, like lysosomal enzymes, that then digest and break down the protein into small little fragments, right? And then another vesicle that is coming from the Golgi that contains MHC2 that was made expressed back in the nucleus, translated by ribosomes into the ER, just like MHC1 and any other protein is, right? Folded up into MHC2, right? But this MHC2 was not loaded up with peptide in the ER, right? It was prevented from being loaded at that point. It was already shipped out of the ER empty, right? And that empty MHC2 traveling in this vesicle now meets up with the lysosomal vesicle that has all these short peptides from the pathogen. Now the MHC2 gets loaded up and it gets moved to the surface of the cell. So let's see what's going on. Back in the ER, MHC2 proteins are being translated, folded up, and ready to go, but you don't want them to be loaded up with peptide in the ER, right? You want to keep them sort of empty, right, or unloaded. So as an MHC2 is made, there's another protein, this green one, called the invariant chain that basically plugs the peptide binding cleft of the MHC2 so it can't get loaded up, right? So now the MHC2 gets shipped out. That invariant chain protein kind of breaks down, leaving like a little plug left, which you can just call the clip. And it's like a little cork plugging a bottle, right? When that MHC2 meets up with vesicles that contain the digested bits and pieces of pathogen that this macrophage, for example, brought in, then the cork on the bottle, the clip protein is released, and now those peptides have a chance to be loaded up. And now the MHC2 has been loaded up with those peptides, these pathogenic peptides, and it can then be brought to the surface. All right. So this cartoon is kind of cool because it compares and contrasts both of those processes, right? So take some time, talk your way through this, right? Good way to study is to just start from the top and out loud, right? Describe what you're seeing here. Maybe start, in fact, with this over here, right? Virus or tumor, a virus or pathogen infecting the cell, and therefore proteins from the virus or the bacteria are in the cell cytoplasm. Notice it also says tumor antigen, right? Later in the semester, we will talk about the important role of the immune system in detecting cells that are turning cancerous, right? That are beginning to lose control of normal cell function, right, and becoming potentially tumor cells. Not surprisingly, in the cytoplasm of those cells, there's all kinds of weird antigens, right? But let's not get ahead of ourselves. Let's just focus on pathogens for now. So talk your way through this, right? And then likewise, after you're done with that side, go to the right side. Here on the right side, you have a professional antigen presenting cell that also gets the same viral or bacterial protein in it, but under different circumstances, right? Because it's not being infected, it is actually actively eating the bacteria or the virus. So again, start from this point, right? And talk your way through, right? All the way till you get to the end where this particular CD4 cell interacts with it. Because if you can do that, then importantly, it's no longer this task of memorizing a whole bunch of things. It's actually piecing things together like a narrative or a story in your head about different events that link together. And if that makes sense to you, then it'll be so much easier for you to remember all the names of all the little pieces, right? I guarantee. Okay, and then this is the last slide. I just wanted to show you, let's start from the middle. Here we have our typical cartoon representation of a T cell receptor on some kind of T cells, that's its upper cell, right? Having the opportunity to bind to some small peptide, the yellow dot here, being presented on MHC1, right, by a cell over here. Right? Now, what I wanted to remind you is the cartoon does a good job of highlighting the fact that the shape of the T cell receptor's variable domain isn't just the right shape to bind the yellow dot, right? Because it has to do that if it turns out that this is the right T cell receptor to recognize the yellow dot. But importantly, just as much of the shape on either side of the part that binds the yellow dot, it has to be the right shape to be able to bind to MHC in order for this presentation to be successful, right? The MHC to successfully present the yellow dot to the T cell receptor. On the left, right, is the same thing, but now more realistically as ribbon diagrams. So on the left part of this upper thing is the alpha chain of the T cell receptor. On the right is the beta. Below is the MHC1 that's doing the presenting. So look, there's two parts to the MHC1, the long part that kind of curves around and goes down, and then the beta microglobulin blob, right? So here it is. So here's the large part that curls around from the right down to the bottom, and then here's the microglobulin blob. Right? So here, the squiggly yellow thing in here, that is the yellow dot. That is the peptide. That's maybe 10 amino acids long or something like that. And what this shows you is the T cell receptor, those variable loops that were made, right? There's 6 of them, just like the 6 variable loops that immunoglobulins have, right? From light chain, heavy chain, each of them have 3 HV regions or CDR regions of highly variable amino acids, right? Remember, T cell receptors are also made through VDJ recombination. So the same process leads to 3 variable loops on the alpha chain, 3 variable loops on the beta chain, and they work together, right? So these 6 loops here are pictured. You'll notice that only a couple of them, the yellow ones, are the ones that actually recognize the peptide. The other ones, the darker purple and slightly lighter purple one on the left, and the lighter blue and the darker blue on the right, those actually are involved in detecting MHC so that this interaction can happen. And if you look down at the figure on the bottom, if you look straight down from the top of the MHC, looking down at the yellow dot, this is what we'd see. So in the space filling model, you can barely make it out. In the background here, right, the yellow thing is the peptide that's fitting in the peptide binding cleft. The 2 sides of the cleft are the purplish colored part of the MHC and the greenish part of the MHC, right? So they hold this peptide in place. And then the outlines actually represent the shadow or the footprint of the T cell receptor coming down on top of this, right? So the black would be the overall outline of the T cell receptor coming down on it. And then the yellow, right, represent the yellow loops seen on the left over here. And then the purple and the blue, right, those are the loops on the left here as well. So you can see that just as much of the interaction here is recognizing peptide, right, as much as it is recognizing the MHC itself, right? And so as we talk more later about how T cells undergo the process of selecting for T cell receptor shapes that can actually successfully do this, we will find out that that selection process is much more involved than the process we learned about in B cells and their immunoglobulins. Okay, all right, that's it for today's lecture. So good luck with this, and we will continue in the next lecture expanding upon many of these concepts to try to hopefully really get these concepts solid in your understanding. All right, bye-bye.